during the next break. Group number 4 will take the lab tour. And then in the next couple sessions, it's kind of like an open workshopping. So the MZ Mind folks will be around to answer questions and kind of help you through exploring the different options, the different tools, different parameters, how they work. They're also going to show a couple more features, but the idea and keep folks online too can ask questions, and we can get some real feedback, dialogue going, and kind of explore what all is available in that tool. I think those were the 2. Yeah. So yeah, I guess I'll continue. My name is Jarmusch again. I'm at the National Institute of Environmental Health Sciences, which is part of the National Institutes of Health in the United States. And I'm happy to be here today to tell you a little bit about the metabolomics research we're doing in the intramural research program, specifically at the NIH. And just as a quick plug and reference, we're actually in Research Triangle Park in North Carolina, which is the only institute that's not in the DC metro area. And I should note that any opinion shared is mine, not necessarily representative of the government's opinion about anything. Just to kind of put people in, give you the scope of what the NIH does and kind of what the intramural research program is. So it's the National Institutes of Health, which is part of the Health and Human Services branch of the US government. And the annual operating budget, at least in 2021, was about $45 billion US, 41 billion euro. And most of that funding actually goes to the extramural research community, which is in support of 90% or so of the US students studying in university, a lot of grants, a lot of things like that. And then the smaller, but still quite significant, 4.5 billion is going to the intramural research program. And that's at least the largest biomedical research institution in the United States. So it's about 1,200 principal investigators, it's 4,000 postdocs, and it's thousands of professional biologists and chemists that work out there. And there are 27 institutes and centers within the NIH. The NIEHS, which is the Environmental Health Sciences one, is one of the 23 that participate in the intramural research program. And the mission of the NIEHS is pretty simple. It's very broadly defined. It's just to describe how the environment affects human health so that we can live better, healthier lives. So you can imagine that there's a lot of different things that we study, and a little bit more specifically about my lab and where that kind of fits in the scope of things. So I'm the director of the Metabolomics core facilities, what we call our lab. We do mostly untargeted mass spectrometry. We work primarily with only NIEHS investigators, but we do work with people and collaborate at universities and across the world. And very quickly, we are a part, and I'm one of the founding members of a trans NIH Metabolomics core, which is another another core in thought, but it's actually like a federated system across the NIH. I noticed that we kind of went a day or two without actually defining what metabolomics is, which I think is good. And I think it's because I take a very broad and liberal view of this. So this is from the European Bioinformatics Institute. It's the study of small molecules, commonly known as metabolites. Again, taking very broad, I think people will be familiar with these 2 metabolites, citric acid and alanine. That's my favorite amino acid, of course. But taking this very broad view, and like what we commonly actually do at NIEHS is study things like your PFAS compounds, right? Things have exogenous origin, arguably, but they're clearly entering the body, so they have the ability to do things. Molecules related to behaviors and exposure, so the nicotine metabolite, for example, and what that may have to do. And if you know anything about North Carolina, in the spring, you definitely need antihistamines because we have a lot of trees and a lot of pollen. There's clearly multiple things that affect the environment of people who live there. I kind of take this kind of perspective in terms of the lab. In name, we're a metabolomics lab, but we actually do exposomics, lipidomics, metabolomics. We really kind of just focus on the measurement objective and not pertaining ourselves to any one singular definition. We are I like to call it discovery-oriented metabolomics lab. It's called untargeted, non-targeted, as you will. And, I'm sure you've heard this before, the fishing expedition that is untargeted. But I show this picture to my colleagues and I'm like, but this is fishing with a net instead of fishing with a pole, right? Like it's a very different type of fishing. And through that, we can measure all these endogenous metabolites that give you an idea of phenotype and function. But you also see these things, as we've mentioned, like PFAS compounds that are related to exposure behavior. At NIH, obviously, we're very interested in the clinical aspect of the work that we do. So we're doing a lot of disease and biomarker research. We are getting more and more into the microbiota and how that influences our health. And a big aspect and focus of the NIEHS going forward will be this idea of precision health, specifically precision environmental health. So how both the physical stressors such as heat in the environment coupled with chemical stressors kind of combine to overall influence our health and the individualized response people have in terms of where they live and what kind of they do. And, I think this has come up multiple times, but this gives you the ability to see novel and unexpected chemicals. We kind of take a biphasic approach to all of our metabolomics research. So we're primarily interested in discovery. So in this kind of analogy, we're looking at the trees, the plants, the animals, the insects, right? Everything in the environment to try to describe what is going on. And then, we heavily rely on statistics and more often, things like RNA-seq, genomic data to kind of help refine our biological knowledge, bring in other measurements to realize that we only need to really care about the ladybug, right? So why measure the entire forest when you only really care about ladybugs? So this is kind of the second phase, right? The validation. And, we are in kind of a privileged spot that time scales and the grant cycles are less impactful to the research that we do. So we have the ability to do these multiple phasic, very long lasting studies to try to really dive into what's going on. And I kind of see this as a spectrum between discovery and validation, obviously. And on one side, you have untargeted, and on the opposite, you have very targeted metabolomics, exploratory versus hypothesis testing, right, in terms of purpose. And we're just going to kind of zip through these, but because I think you all have a general perspective, but just to catch up anyone, the coverage in untargeted is pretty extensive, hundreds to thousands of chemicals. With targeted metabolomics, you tend to be much smaller, right? Panel type of methods or those kind of pathway assays are definitely kind of fall in between. The outputs are peak areas, targeted its amount or concentration in terms of fixed amount. The variables we call features versus targeted, which we get out the specific chemical names, right? So I would argue the primary technical challenge in untargeted is the feature annotation bit, which is why I think it's such a focus of multiple tools today. And also the concurrent chemical measurement, right? Like you're trying to measure everything at the same time. So there's going to be interferences, there's going to be complexity, there's going to be shoulders on peaks that maybe you need to resolve. Whereas targeted, you're going to spend a lot of time really developing the method, making sure it's as accurate, as precise as possible. To decouple that from the biological interpretation, the untargeted is really hard to understand actually what's changing, right? You have all these statistically significant features, but then you have to piece it together to actually understand what's going on in the system. And a lot of my biology and medical doctor colleagues, they don't really want data. They're more interested in the answer. And then they look to me to like, how do all these things piece together? I'm like, I don't know. So it's kind of hard. Whereas in the targeted, you have to have some idea about what to measure up front. And along with that, we kind of mentioned different types of instruments, but they kind of also fall along the spectrum. You can argue where their position goes, but this is kind of how the instrumentation in our lab kind of we utilize it. So we tend to use a lot of Orbitrap systems for the discovery phase. And we have a Bruker timsTOF Pro that has imability. It's a QTOF instrument. We kind of use it as a middle ground. We do some validation work. We use the imability aspect to really help resolve peaks. It's like, is that really one feature, or is it two convoluted structures that we just can't resolve chromatographically? And I'm personally not very good at chromatography, so I heavily rely on the imability to help us out. And eventually we'll have a triple quad, but we're waiting on some lab renovations. We're going to zip through this real fast, but I thought it important to bring this up and over again. So you have a sample, you put that into the chromatograph, and then you're doing some kind of affinity or interaction-based separation over time, right? And then you send that directly to the mass spectrometer to measure the mass spectrometry lines. So you get this type of data structure, at least in reverse phase chromatography. You have the most polar things coming out first and the least polar things coming out second. And then on the MS1 level, we use that to assign the molecular formulae and also predict, predict the molecular formulae and look at the isotope patterns. And then the MSMS, we isolate that either in space or time, as mentioned. We apply some kind of energy to that, and that imparts this fragmentation that relates then to structure. We take again, kind of a data-driven approach to our untargeted. And I think Kirsten in the lab put this perfectly. We're trying to measure what matters instead of everything that's there. So I think Robin maybe mentioned it. It's that comprehensiveness versus what is actually statistically significant. Again, the collaborators are looking at answers, not necessarily characterizing the entire chemical space. We do a little bit of that. So everything is a feature. We do the annotations via MSMS database match, very similar to what you heard. We do data cleaning, and just to add another layer of distinction, you can do that based on biological information, or you could do that based on the technical measurements. We tend to rely and filter only on the technical aspects, not applying any known biology to it. And then we do statistics, and again, kind of branching out from something that you've already heard, you're focusing on the significant things, and then of those, what are annotated, looking at what we can identify through, retention time matching, re-annotating, and then the hardest bit is when it's unannotated, and then we're going to talk a lot about what you do when you go, when you have that situation, molecular networking, formula prediction, in silico tools. It's very much, these are not words I use, and I avoid them at every grant and every meeting. Comprehensive and optimal are not characteristics of untargeted metabolomics, I would argue, because it is a small chemical space compared to everything that is actually there or possibly there. So while some people in the field are striving for harmonization, I think harmonization is good, but we can't be all doing the same thing. Otherwise, we're all measuring the same space. So I think we really do need like an ecosystem of methods and strategies, but we actually can interrelate and understand all those things together. And you can apply some heuristic knowledge to kind of limit what you're measuring. And then I think I would argue that it should be a biphasic system, that we should always do some discovery and then validation, particularly on the biomedical side. So I have some examples to share with you of things that we're actually doing. I apologize, this is by no means comprehensive to actually what we do, but these are things that I could share with you pretty readily. And I think that kind of display different aspects of what we're doing. So we do a lot of basic biology. So this is cells. So this is understanding how the yeast metabolome responds to oxidative stress. And potassium bromate is particularly of interest in this case, because they actually add that to bread in the United States. So the question is like, are we constantly exposing the entire population to this oxidant, right? And as we go through these yeast cells, and hydrogen peroxide is a known oxidative stressor, you clearly see a metabolic shift. So this is analyzing our metabolomics data using PCA. So the blue points are definitely not like the green points. And one of the principal features driving this to reduce it down to the univariate statistics is 8-hydroxyguanosine. So this is a known marker of oxidative stress to DNA and RNA, right? But it's not the same between potassium bromate and hydrogen peroxide. It's actually differential in some way. And what the collaborators have actually gone through is characterizing genetically the predisposition of which actual mechanisms are used to compensate for these oxidative stresses. In this case, glutathione is also depleted in the potassium bromate quite significantly. The depletion of glutathione can be related to, well, in this case, it can induce apoptosis in cells. So this actually made it be a very much not a thing that you want to actually do to your cells. This is another study that we participated in. Again, kind of different things. So this is moving to now CD4 positive T cells. So this is a human line of cells that are within the lung. And specifically, this is a collaboration with NHLBI, heart, brain, and lung, I believe. They're all very confusing, even for me. And basically what they did is they wanted to understand what the effect of this arginase 1 was in this model. And if you actually knock out arginase 1, what they were actually showed is that you actually reduce the lung damage caused by these immune cells. And they wanted to do untargeted metabolomics to see if they could characterize any other pathways or what else was going on. And in fact, we saw a depletion in glutamine. So the idea is that there's kind of a metabolic difference in a shunt that it's actually imparted by this knockout as well, that may be related to different disease mechanisms. And again, this is a very pictorial way of showing it, but in the right-hand side, you are reducing the amount of lung pathology by shortening the period of time that these T cells are present. And another example of kind of some very basic biology is cancer cell lines. So this NCX4040 is a chemotherapy agent. And they knew that it was killing the cells, but they didn't understand which mechanism. So they went through and we discovered that it was imparting differences to the acylcarnitine pathway. Specifically, it was inducing baroapoptosis, which is a characteristic of programmed cell death that differs from apoptosis, different mechanism, same result. But it's this was recently published. And then if we move a little bit stepwise to a bigger system, now instead of cells or tissues, now or cells, let's talk about tissues. So this is now understanding a novel gene that was identified at NIEHS called LIS3. And what they realized that people who have mutations in this tend to develop polycystic kidney disease in humans. So a mouse model was developed by the collaborators, and we obtained the tissues for a wild type and knockout condition. All of the knockout mice develop polycystic kidney disease, so it's a very reproducible phenotype. We did multiple animal models or animal replicates. We subsection the kidneys because they are heterogeneous. I was very excited by the top trace because we had not done really any tissues. So some of our first examples of this, and the collaborator, I was very happy, and they were kind of confused, like, what's this trace even mean? This is meaningless to us. And then we started pointing out, the extracted ion chromatograms, and all of a sudden they became very excited, right? They're like, oh, wow, you can do a lot with metabolomics, right? I was like, yeah, and then, again, kind of analyzing all the data together with PCA, and you see a clear differential between knockout and wild type components. Reducing that down to singular kind of molecules. We found that citric acid was actually increased in the knockout. And these are very dramatic increases, right? In citric acid, succinate is actually lower. And this is kind of counter to their hypothesis. They knew TCA was dysregulated. And what they ended up showing was that there's a shunt from glutamine into the pathway, and that's actually what's causing the uptake in terms of citric acid levels. And this is kind of a cautionary note, but we also saw these features that were differential. And this is getting to like understanding the biology and interpretation is complex. So indoxyl sulfate is a known uremic toxin, known gut metabolite, and increased heavily in the knockout, as well as this, I think this is isoflavone. It's found in soy. The mice are fed a lot of soy products in terms of their chow. So we traced this kind of back. We did a cell model that kind of reaffirmed the citric acid, but refuted these particular molecules. And what we basically were able to reduce this down to and correlate this with renal function. So like when it comes to like tissues or biofluids, you know, it's more complicated metabolically. You really see the superimposition of both the underlying metabolic changes as well as any kind of functional changes that are occurring at the organ or organism level. So a little bit of a cautionary note. Speaking of whole organisms, this is like the next level up. So we can do whole Drosophila. So we were doing a study between looking at the conventional microbiome versus germ-free, and then tracking that over the age of these flies. Two kind of novel questions and developing the model. These are folks at NIEHS. So we found this particular feature of butyryl carotene that was dysregulated in terms of the germ-free, but also displayed some trend over time. So we tracked this down. It seems like this is related to the butyrate produced by the bacteria that is actually changing some of the butyryl carotene levels. And then you have other things that are clearly more related to the age of the fly. So this is the methionine levels as they age. And just kind of orient people, I don't know how many people are fly experts, but 3 to 10 is kind of like they're growing up, they're maturing. 30 to 56 or 30 to 45 is kind of when they're in adulthood, and 56 is kind of when they're in later adulthood. So as the flies are actually developing, they're changing over time. So Drosophila is a very common model of a lot of things. So it made our investigators really think about where they're selecting for time and how they do follow-up studies with all these things. And just a couple short notes. We don't just do a bunch of applications, right? We actually commonly end up developing a lot of different methodologies, some of which I don't have time to share today, but you know, we're asking a lot of unique questions like, hey, I want to study the effect of the smallpox vaccine, but I have mouse ears. Can you do metabolomics on that, right? So I could probably talk about an hour in terms of everything we tried, the optimization of that. But it's a really interesting place to do research. We recently published a paper on a different method of doing data cleaning. So this is taking a pooled quality control. So that was mentioned by, I think, Philip number 2 yesterday. And he is specifically a small aliquot of every sample goes into that. And here we're looking at kind of the fundamental response to an increased amount. So it's not a dilution series. We're doing different volumes of it. We're injecting different volumes of that material over a very narrow range, and then kind of looking if there's a fundamental response that we would expect. I think someone will probably mention GMPS dashboard. I thought I'd throw this up here because I think this is a really good collaborative tool. But this is a website that you can go to. You can drag and drop files, and we use a lot for doing like kind of collaborative, looking over the data, reviewing it, because it has this share or this follower function, right? Sync follower function. So you can actually show the collaborator remotely exactly how you produce an XIC for whatever molecule you want, it's very helpful in terms of communication of the data. And then just to plug this, I think Ming will probably talk about this a little bit later, I think on Wednesday. But the mass query language, or MassQL, This is a specific query language. It's related to SQL in terms of how you write it, but it's fundamentally you can perform queries on whatever data that you have. It's vendor agnostic. You work from .mzml files, as we mentioned with the mzmine. And what this does is allow you to ask a lot of complex questions about the data that are kind of hard to do in general tools. So you can start to ask a question of like, where do I find a sulfate product ion, and I have a sulfate isotope, and that sulfate also contains a carboxylic acid functionality, and it has this retention time window, kind of, because I kind of know something about the polarity. That's kind of a hard thing to do in a generalist tool. So it's kind of unique, and I'll point you to the bioarchives paper, still in revision. But I think there's a compendium. So I think there's like 150 pages of examples and different things that you can do. So the idea is that you can write a query, you can share it and upload it to that compendium, so that if someone else wants to write a query for looking at PFAS molecules, you don't have to necessarily know how to write all the different things. You can just look that up, actually reuse it on your own data. And I think this will be natively supported in a bunch of different tools, including mzmine, right? Yep. And I think it's R and Python and MS-DIAL. So there's you can use the same kind of query in multiple locations. So you only have to learn arguably one way to do this, and you can use whatever your favorite tool is. And with that, I'll leave some time for questions, but I also thank the most important people, just the people in the lab, Kaylee, Kirsten, Heather, who are the chemists in the lab. And if you have any questions about how the government system works, I'm happy to address those. The collaborators, Ming specifically, because he helps me out all the time. And Dylan is our new contractor in the integrated bioinformatics core that we do a lot of collaborative work with. So with that, I'm happy to take questions. The potassium bromate. So the question is if it's dangerous if you're exposed to too much. So it's still in yeast. It's a long process, but the idea is first to establish kind of the fundamental biology. So there seems to be epigenetic changes that are associated with it, and that's more likely than to be applied to all the blood samples that we have and try to track to see if, like, what is the unique signature across the entire population, rather than doing a very large untargeted metabolomics study. So that's a good question. So the question is, why is there potassium bromate in bread? And the answer is, one, I don't fully know. I asked the same question, but I think it's used as a preservative, which there's definitely different rules in terms of the US in terms of what you can add. It's probably a preservative for the same reason that it causes oxidative damage, right? Probably disallows certain things to grow. Back to mzmine.